I decided to start my talk in biotech style, which is just choosing 3 words that really describe what I'm doing. The first of the words is electromicrobiology. This is a relatively young subfield in microbiology, and the term was coined by my colleague and friend, Yuri Gorby, who already in 2009, when we were starting to bring to light some of these discoveries, he had the visionary comment that he thought we now stand at the edge of a new scientific frontier, and he was right about it. And this brought many challenges, but also many opportunities for education that I wanted to really capitalize on, and I'm going to share with you. But surprisingly, also for entrepreneurship, and I really didn't plan this. It was a scary path, but it became intertwined with education as well. So I always like to start my scientific talk with electrons because this is the time in which half of the audience is looking for the nearest exit. We fear talking about electrons, although they drive life, and the best person to acknowledge this was the Nobel laureate, Albert Szent-Györgyi, who gave what is, in my opinion, the most accurate and the most beautiful definition of cellular life. Life, he said, consists of electrons looking for a place to rest. He was referring to the restless electrons that really energized the first reactions in a hot early Earth 4.5 billion years ago, when life couldn't have existed yet, but the first building blocks of life were created. And from these reactions, restless electrons were moved to catalyze the synthesis of the first biomolecules. Here you have the RNA synthesis that is proposed to have occurred thanks to these restless electrons on the surface of clay minerals in a hot early Earth. And here we have the emergence of the first self-replicating molecules and the first similarity to one of the major characteristics of cellular life. Fast forward 4.5 years of evolution, and what we have is a diversity of processes enabled by microorganisms in most creative way, but still moving those restless electrons to power, growth, and reproduction, motility and communication, so they become cosmopolitan and they colonize all places on Earth. And we now know that there are many microbes that do not keep those electrons inside the cell. They actually zap the world around them. They really transfer those electrical signals to the outside world and to each other. And it's such important, the impact that collectively they have on planetary processes, that we now know that there are global implications for these activities. So I was very fortunate as a postdoc to be witnessing the birth of this subfield in microbiology, studying a group of organisms called Geobacter, which is a Latin name that means bacteria from Earth. And this is telling you that these are really very ubiquitous organisms because they have evolved a very unique mode of respiration. They don't respire oxygen like we do, they respire rust, iron oxide minerals. And this is very challenging because they have to zap those respiratory electrons outside their cells to really get the mineral accepting them. And what they do is they actually cycle the iron that is trapped in the minerals. They solubilize some of this essential metal for other microbes, but they also make a magnetic mineral called magnetite. So the respiratory mechanism that enables this process really is universal, and you find it in many bacteria that you may be more familiar with. It all starts with the metabolic reactions inside the cell that harvest those electrons with protons in what we call reducing power. In this case, NAD, the carrier molecule. And then there are dedicated proteins like this NAD dehydrogenase that are actually separating those protons and electrons to create the proton motive force that generates energy for the cell. All comfort zone for us at this point. These electrons, however, get stuck in these lipophilic compounds of the inner membrane called quinones and need to get out to make an electronic connection. And this is what is unique about Geobacter. We know that is loaded with cytochromes, a type of metalloproteins that use the metals to really move those electrons fast. And these are just some of the ones that are conserved in all Geobacter that have been identified. There is something very unique about these ones, is if there is no electronic connections between the cell and the mineral, they are going to keep producing some of the cytochromes, and they are going to continue to store the electrons. And this is one in electronics is called a capacitor, cells that work as capacitor. What they do is they can accumulate up to 1 million electrons for each cell until they manage to zap the mineral outside. So in case you don't believe me, this is the type of cultures we grow in the lab. The red color is really the amount of cytochromes that they produce during respiration. What we know is that there are ways to breach the insulating barrier of the outer membrane also with cytochromes. Some, they are alone, some are associated with porins. And early on in my postdoc, I became intrigued about this localization of these cytochromes. I always envisioned the cell like a battery, maybe a positive pole and a negative pole. And I was frustrated because every single outer membrane cytochrome that I tried to immunodetect, in this case is one called B, it was all over the cell. And this was very conflicting for me because when I look under the scope, I did not see the whole cell buried by these minerals. What I saw was pretty much naked cells, and I don't know if you can appreciate some lateral extensions, are the ones that are coated with the iron oxide minerals here in black. And when I look closer, I realized that there was a network of filaments that were enabling that electronic contact. And I took a genetic approach to actually show that these were pili, which is another Latin word that means hairs. So these are hair-like filaments that most pathogens actually make. So they are really well known. But there was something unique about these bacteria that were using those pili for respiration. And this was the beginning of many years of work that I want to summarize for you in just a simple slide. At this point, we knew that there has to be a change in our model of respiration in these organisms to include the pilus fibers. And we found in the genome, and we proved this genetically, that is conserved, as you find in other bacteria. And this is a load of proteins that really make sure that a single peptide, the pilin, gets polymerized, and there is a fiber that protrudes outside. Nothing new here. The difference is that this pilus is immersed in this highly charged environment. And it conducts the electrons outside the cell, and we didn't know how this could be possible. It was a paradigm in protein electron transfer. So we identify how it protrudes, and then we found that once the pilus charges are actually passed onto the minerals, and the pilus remains trapped in its own reduced product, the magnetite. And I always wonder, are they dying after a single breeding cycle? It doesn't make sense to get rid of these minerals. And they do so by actually depolymerizing the peptide, retracting the pili, shedding off the mineral, and starting all over again. And this actually helps them in doing more reactions. We found that actually this pili can zap uranium, a soluble radionuclide that could be very toxic if it penetrates inside the cell. So the pili actually shield the cell, they bind it very effectively, and they zap it and mineralize it to protect the cell and also gain energy for growth. So it's a double strategy that they have. And in case you don't believe me, look at this. I tried to colorize in yellow the very few filaments that were visible in all this uranium mineral that was precipitated. And we could genetically engineer more pili, and it had a more positive effect in the capacity to bind and reduce uranium. So the question that lingered here was, okay, we did everything that a microbiologist with our tools and our knowledge could do. But the answer that we were missing is, are the pili nanowires? So I did what every reputable scientist will do when confronted with a completely new field. I Googled. And I asked the question, what is a nanowire? And I got something like this, a nanoscale rod of semiconductive material. Then I stumbled in the word transistors. Say, okay, next phase, Google, what is a transistor? And this is when I realized that things were getting complicated. It was a little bit over my head. And if I wanted to answer this question, I had to plan a strategy to start educating myself to start with. And this brings me to the second word of my talk, education. I had to educate myself before I could plan what to do next and how to mentor my students and postdocs in this educational adventure. So let me give you a little bit of summary of what I did. To educate myself, I started reading and learning what many of my colleagues had really discovered about bacterial pili. And it turns out that it provided very interesting information. What you see here is a structural model of the pilin subunit, the peptide that makes the pili of Pseudomonas aeruginosa. If you are familiar with cystic fibrosis, you know it's a very important pathogen. So this is a good representative. And it has a modular structure that is conserved in all bacterial pilins. So it was a great way to start. And you can start to see that it's almost like a lollipop. It has a helix, that's the stem, that's the one that assembles the peptide into the pilus fiber, and it has a globular head that engulfs that helix and is the one that gets exposed outside, and it allows this pili to function in attachment, in surface translocation, etc. So that was good. So then I built the first of my model. I had never in my life done any type of computational modeling, so my first tools were very rudimentary. This is like the fine-tuned process 12 years later, and I found something very unique, that you still have that helix that is essential for assembly, but pretty much everything else is gone. You just have that little random coil exposed outside. So they are short, they are very short, but they also have aromatic amino acids, many. High aromatic density, and that mattered because when I started reading literature in electron transfer, I realized that there was many papers that are out there that were talking about peptide electron transfer. Engineers and physicists have been studying them for a long time, and I was just as a microbiologist catching up. And they knew that aromatics were very important to move restless electrons. So things started to make sense. Thanks to this, I did what everybody should do, is to collaborate. And I actually was able to collaborate with an experimental and a theoretical physicist. Stuart Tesmer was the one doing the experiments in the more physical aspects of the work. And Gustavo Feliciano is a wonderful collaborator in Brazil that does all the theoretical modeling that was needed. And I'm going to give you an example of really my favorite story that came out of the synergy of our interactions. And it involves my postdoc, Sanella. Sanella was a physicochemist. She had always bought proteins from Sigma. She never saw a culture of a microbe. We had to teach her. And she actually ended up developing a wonderful method to purify the pili, but to purify it under the standards that physicists wanted for electronic measurements. Absolutely not even a trace of redox organic cofactors or metals contaminating. And she reproducibly managed to do that and have very clean samples. And then the student Josh Visi used a microscope that is nothing I had really imagined I would use, a scanning tunneling microscope that produces an image of the electronic structure because it's shooting electrons and receiving electrons to measure the conductivity of your sample. So this is what we found, a beautiful pilus fiber that is not your biochemical structure, but is actually the electronic structure, with a lot of detail of where those restless electrons really are. And we use as a negative control the Pseudomonas aeruginosa pili that Sanella purified. Believe it or not, this is a pilus fiber because it's just so insulating, as you see in this flat line in this quantitative plot, that you actually have to increase the voltage so much that you're shooting electrons at such high voltage that it creates instability, and this is the best image you can get. But look what happens with the conductivity plot that we saw in the Geobacter pili. We saw that it was conductive at all times, even when you don't apply a voltage. So this is a structure that has this remarkable ability to have restless electrons even when there is no voltage. So this was very intriguing. We decided to focus on the biologically relevant voltages, and Kathy Walsh, a student in physics, froze the sample. Our idea was, can we freeze those restless electrons in place? And we did this because we could really get a lot of mechanistic information. And you see that we actually stop those restless electrons at the biological voltages that really matter in the respiratory chain from the inner membrane to the outside minerals. And this is important because it gave us information that those electrons were moving because they were actually hopping from one relay station to another. And that relay station, I always wondered if it could be those aromatic amino acids. So this is where the computational model made a huge difference. This is an atomic resolution computational model of the pilus fiber that my collaborator, Gustavo Feliciano, built. You see one of the subunits to get a sense of a scale. What we're talking about is a nanorod that is about 2-3 nanometers in diameter. But look what is hidden underneath, this beautiful path of aromatic residues that actually have interaromatic distances that are reported to be ideal, optimal for electron hopping. But if you look closer, you have to look at the geometries. Each one is going in different angles. And this also provided very important information because it confirmed that we had this multi-step hopping in contrast to a metallic conductor that actually has those rings very, very close to each other with a rigid geometry so they never misalign. So from here, I was bold enough to ask my postdoc, let's build chips. And let's really show that these are nanowires. And Sanella was just wonderful, and she learned to microfabricate these electrodes in which we have an insulating layer of silica oxide, and we actually pattern different shapes of gold electrodes. And our idea is let's deposit the pilus fibers at the edge. And then let's use these conductive tips in this wonderful microscope to shoot electrons and measure charge transport. So she did. It took 2 years to get this beautiful picture, but she really developed a beautiful method to reproducibly measure charge transport. This is how 13 different pilus fibers from 4 different biological replicates conduct electrons. What this means is, this is something that physicists always ask when I write papers in the field, charge mobility. We were able to calculate quantitatively how easily those charges move. And just this is something that microbiologists don't care much about, but the important thing is that it was actually comparable to redox polymers that are actually being considered for interfacing with electronic devices, solar panels, for example. So this was really cool. But what matters for us microbiologists is that we demonstrated that each fiber was transporting 1 billion electrons per second at the very low voltages between the inner membrane and the mineral outside the cell, which was remarkable. Just to give you an idea, a cell during respiration, the metabolism is only spitting out about 10 million electrons per second. So what we did is use these computational tools to actually make like a skeleton of those aromatics in the simulations coming close together. So in red, you see the hot spots with those aromatic dimers get very close to each other. And then what we did is find the aromatic amino acid that was responsible for half of those contacts, so we replaced it with an alanine. And then we created this mutant, tire 1, and when we got the pili out of them, we got magic. This is exactly how the genetic engineering helped us demonstrate that we actually had found the path for those restless electrons. And then this opened many opportunities to really manipulate. Can we make better paths? Can we control the properties of the paths, for example? Something else that we got out of these modeling efforts is the fact that we were able to identify the metal traps. And there are these red areas that each of the peeling is contributing to decorate the surface. These are negatively charged amino acids that actually provide a beautiful cage for any cationic metal to be bound. And then they position it very close to a tyrosine to be able to transfer the electrons as well. And my student Becky got really motivated to start doing computational work. And what you see here, she developed a model docking the uranium atom that we had experimentally shown to be relevant in vivo. And this is a beautiful example of how experiments and computational models can really help each other understand the biology of a system. Because using a spectroscopic analysis, we had modeled the actual configuration, the actual atomic environment of the uranium in these samples that we had prepared before, and they match exactly, even the atomic distances, to what we had predicted in the model. So this opened many opportunities for us. What we did is we got funded by the Department of Education to have a new graduate training program in bioelectronics. And I'm going to tell you some of the work that 3 exceptional graduate students did to contribute to the success of this program. Becky and Krista in my lab. Angelina's was a chemical engineer that I co-mentored with a colleague in chemical engineering. Becky, genetically engineered recombinant peelings that we could predict had all the ingredients needed to have that type of conductivity once assembled. Angelina's, as a chemical engineer, develop and optimize all the recombinant production so we could really manufacture as many of these building blocks as we could. And Krista made magic happen by actually developing the tools to integrate proteins into the conductive materials and electronics. And I'll give you just some examples. We follow the protocol of getting a patent first and then getting the paper out. So it's a very highly designed approach to mentoring to help the students. I compare it to buying a Christmas tree and then decorating it as much as you want. That's the process. So this is an example where we got a patent for these biocoatings in which we actually interface with genetically engineered recombinant peelings and develop in vitro planar assemblies. And the beauty of this is that there is a lot of interest in using them as biocoatings because in prosthesis they prevent corrosion and they actually promote the integration during implant as well. But from an environmental point of view, we also know that they have more of those metal traps concentrated. And now we can interrogate whether we can use other nasty toxic metals that the cells cannot tolerate by themselves. And we can develop novel platforms for bioremediation as well. And this matters, and in these times matter because we are witnessing a deregulation in environmental policies. And I think as microbiologists, we have the knowledge to provide solutions, but also the moral responsibility to inform of policies for restoration and prevention of this pollution. Then we did something, the paper will be coming up in the next couple of months. We actually now can make synthetic protein nanowires that have exactly the same conductive protein properties as the native ones, but now we can make them in quantities that enable really interfacing and making bioelectronics possible. And then today I don't have much time to tell you about the work that we have been doing in parallel to actually use these nanowires to interconnect cells and make electroactive biofilms that are on an electrode so we can harvest the electricity from them. And these are very interesting because they can process a lot of organic waste that industrial plants actually find a nuisance and they want to get rid of it or maybe harvest energy that is wasted otherwise. So I'm going to tell you into the entrepreneur path that this type of experiments really led to. And I'm going to focus only on our work to making bioelectrodes and the applications that we found. And Becky was a student that stayed for a postdoc to really propel these commercialization projects. And Bouchang is a chemical engineer. And here you see again the pairing of 2 different disciplines to really make the project advance. And this really led to the startup company Bioelectrica that is still a very young company, but it has become really a motivational force for postdocs that didn't see a future didn't want a future in academia. And it has attracted tremendous interest even in students to really have this type of training that can really open new career opportunities for them. And what we basically do is we retrofit just about anything that is available in industrial plants. We figure out what are their needs in terms of waste, and we use microbes and bioelectrodes to really clean up the waste and develop kind of harvest energy for the process in the bioproduct, for example. So I'm going to give you 2 examples. The first one is actually Bouchang developed this platform. It's a reactor in which We had a fermentative microbe that was like a garbage disposal. It loved all the waste from industrial partners that it could do. And it produced ethanol, but it produced waste byproducts that we simply could not process. And this was really not a cost-effective platform, because just getting rid of a purification protocol of all this waste would have been too much. So we developed a bioelectrode that was specifically custom tailored to remove this at the same speed as it was produced. So we stimulated ethanol production, and we clean up the broth. And this is what the video shows. Bouchon got a Spartan fan to just show just how much electricity. If we can play the video again, you can just see this surprised us, because we have a bioelectrode that is half the palm size of my hand. So we were producing a lot. And this was a failed experiment, because what you cannot see is the message, is the punchline: go green, Spartans will, which is the motto of our university. So we developed other platforms thinking, okay, we have really technologies that can address the treatment of very dirty waste that otherwise wouldn't be treated in any other way except being sent to a wastewater facility at a price. So we actually developed a platform with human urine, and Bouchon actually contributed with his own brew on a daily basis to really have this platform. The bioelectrodes performed beautifully. We had to do some adaptive evolution and genetic engineering of the fermentation. But the bottom line is that we found a platform in which ethanol is produced as a sanitizer in low concentrations, enough to prevent the growth of organisms. But at the same time, we're cleaning up the water. And we thought we got funds from the Gates Foundation for this platform to think about third world countries to promote water reuse and stop the big problem of sanitation with all the complications, health issues that come from that as well. Oh, and if you could go play the video again, this is the experiment that worked with the fan, because we could actually see our university near Michigan State working there. So I leave you with this, nothing like ending a talk with a toilet, because we really cannot understand the needs of third world countries in a world that has sanitation, wastewater facilities. But there are efforts to really have even toilet discoveries. The Gates Foundation compared the design of efficient toilets for third world countries as the same effect that having fridges made in really prevented food spoiling and really disease that were transmitted through food. And there are very inexpensive toilets that are used as community latrines in many countries. This is actually projects in Kenya that the Gates Foundation is funding. And there are toilets that divert the urine, so you can actually have very inexpensive treatment methods to just use this type of sanitized water for flushing, or really use it even for agriculture. And women play the key role in those African societies to sanitize and to do agriculture. And it's a very nice way to empower women in societies to really reinforce their role as the breadwinners are raised. So it has many social implications. So with that, I want to tell you that I do have a postdoc in which I'm going to provide training in commercialization, patents, etc., and business training. And if you follow me on Twitter, I will be posting the link for applications in about 2 weeks at most. So please do apply, and don't fear it, be brave. And I say this especially to women, because we usually, if you don't qualify 100%, you don't apply. Please apply, surprise me, and join me in this adventure. Nothing can go wrong. And with this, I leave it with all the acknowledgments. You may have noticed that I have been very fortunate to attract many women. I think there is power and many women really joining these challenging projects and interfacing with disciplines where women are traditionally underrepresented. And I really owe all of that to them and to their contributions. I think I'm going to leave it just like this with the acknowledgement. Thank you very much.